Welcome back. We're now ready for our questions. We have some questions already phoned in, and we'll get to those in a moment, but I wondered if anybody on the panel would be interested in offering a comment or a response to someone else's. Yes, go ahead, Dr. Kevlis.
I want to take issue with some claims advanced by Dr. Recksteiner. I must say, by way of preface, that I'm not a member of the Human Genome. Is this on? Or? I'm not a member of the Human Genome Project and have no funding for it or anything like that. I am a friend of it, though, because I believe that it.
We want to hear you here.
Okay. I wanted to comment on some points made by Martin Recksteiner, and I want to preface what I'm saying with the notation that I have no official connection with the Genome Project. I have no funding from the Genome Project, and that I am a friend of the Genome Project, however, because I think it's a valuable and important scientific and medical endeavor. The points I want to comment on are concerned the distribution of resources in the Genome Project. In 1991, the budget, the actual expenditure of funds on the Genome Project took 1.5% of the NIH budget. It took only 3% of all external grants given by the NIH, which hardly seems to me to be an enormous expenditure of money by any measure. Secondly, with regard to the point that only 10 people were funded at $20 million apiece, the fact of the matter is that the Genome Project in 1991 funded 175 different projects at an average grant size of $312,000 a year, which is higher than $200,000 a year, which is the average grant. But again, it hardly represents a major concentration of funds. There are 8 genome centers, to be sure. One of them at least is getting roughly $3 to $4 million per year. But even all of these centers house multiple individual projects. Secondly, apart from just throwing in some factoids as monkey wrenches here for comment, not so that these things don't go unchallenged, it seems to me that with regard to training, there is a romanticized view of just what biomedical research has consisted of in the past and even what much of it consists of now. A great deal of biomedical research, even without gene sequencing and gene mapping or whatever, like all of research, consists of 99% or some large percentage of sweat and several percentages of inspiration. There is a lot of drudgery in research, whether it's scientific or historical. It seems to me there's a romanticized benchmark as to what research without the Genome Project will look like. And I would make the claim that given that so much of the Genome Project has been devoted in recent years to the development of technologies for doing research that is now extremely tedious, such as sequencing and mapping, that you will find that it will bring indeed a liberation from such drudgery rather than imprisonment in it. The history of recent biomedical science, and I mean since in the last 50 or 60 years, shows that the increasing incorporation of technology into biomedical science has liberated people much more than it has imprisoned them. And so it seems to me that this is, from the point of view of the practice of biomedical science and wise science policy, that these are issues that need to be kept in mind, whatever you may think of them.
I would like to add one point of science to that argument. Let me answer first, otherwise I'll have to answer so many that I'll forget what you said. So I've already got, there's already 5, if I've got it right, about 5 different issues raised here. One is what percentage of the budget is it? And in 1990, the numbers I have, in 1990, I don't have the 91 figures on the disbursement of cash, $50 million direct costs, and almost 40% of it went to 10 investigators. I didn't make up the numbers. I added up all the genome. I don't have the 91 figures. Perhaps it's a little better in 1991. I might also add that although 175 grants were given, in the ethics area, the grants are of the order of $20,000. At least in 1990, there was a marked asymmetry in the distribution of money. In fact, the 1990 budget wasn't 3% for the genome ethics. It was 1.5%. There were lots of grants, but in fact, most of them were $20,000. We had at one end lots of grants at $20,000, and at the other end, a few grants at $2 million. Well, that's a fact. I mean, I've got the book in my room. Anybody who wants to dispute that, we can go count the grants and count the money. I don't have the 91 figures, so I can't address those, but I would urge you that maybe this average of $300,000-$400,000 is $2 million plus a lot of 20s, and you can get a pretty good average, all right? Second, we have 3% of the budget dictated now in NIH through the efforts of a single senator, I would claim, Domenici. Imagine if this becomes a style. There are 100 senators, and pretty soon we have 300% of the NIH budget mandated through the efforts of politicians, okay? So I would argue that, well, 1 or 2% here, 1 or 2% there, pretty soon we may in fact have a totally pork-barreled NIH budget. I don't believe that that's good for the US scientific establishment or for our country or for medicine, all right? Drudgery. In fact, Sidney Brenner, another advocate of the program, of the project, an English scientist, claimed that sequencing ought to be a punishment for crimes. The amount that you had to sequence was proportional to the crime. All right, so a megabase was petty theft. Yeah. 10 megabases was assault. All right, but in fact, sequencing is drudgery. All right. Finally, machines. The project is sold as a great way to generate a lot of machines to make life wonderful for us. I would argue that there are already commercial companies, ABI, Milligen, who make machines. And in fact, they have all the reason in the world to make better machines. If they make better machines, they make more money. I don't see that the NIH should be paying for product development by private firms, all right? And I think I've covered 4 of your points, and I'd be happy to cover Norton's.
Can I say something about the NIH budget?
Speak directly, they tell me.
Oh, okay. The NIH budget is not so easy. That's how you can look at get these statistics, which I'm sure are true, as opposed to these statistics, which I'm sure are true. The extramural research has to be separated from the intramural research. And of the extramural research funds, which goes out to academic universities, more than 50% of it goes for indirect costs. And so that's really not counted. If you calculate it and you subtract the amount that goes for indirect costs, and you look at the direct cost of the Human Genome Project, I'm sure it comes out to much more than 3%.
Okay, we have a question from DePaul University in Greencastle, Indiana for Dr. Ecksteiner. Why wait to sequence the human genome until we can understand the physiology of the cell? Why can't we do the research now, and when we uncover more discoveries about the physiology, combine the information right then and there? If we wait to sequence the genome, then when we do understand cell physiology better, we'll have to wait longer to combine the information of the 2 disciplines.
No, I don't know if, can you hear with this mic?
Talk louder.
All right. Okay, I would argue that important pieces of biological machinery are being sequenced independent of the Human Genome Project. I, in fact, in my lab, have sequenced cDNAs for components of machines, biological machines, that I'm interested in, all right? In fact, I do DNA sequencing, so I'm not a throwback who thinks that everything in the future is bad. I'm not even against DNA sequencing, all right? I do it in my lab for specific problems. Those who are interested...
What was your crime?
What?
What was your crime?
My crime was being interested in ubiquitin metabolism,
Well,
all right.
Good.
So is it... All right. So I would argue that, in fact, we should let individual investigators who are studying components of cells, presumably of biological and medical relevance, sequence those pieces of genes that are important to them. In fact, it's being done all the time. The databases were increasing, by Brenner's account, at tenfold in 5 years before the Human Genome Project. Why? because a lot of people are sequencing pieces of DNA, relevant pieces of DNA. 95% of the genome, I claim, is irrelevant. And we should not spend money sequencing irrelevant pieces of DNA when there are individual investigators, myself included, sequencing what they consider to be relevant pieces of DNA.
Well, your criminal sequencing of DNA is costing us $10 a base pair, and Solstrom is now sequencing DNA at 50 cents a base pair.
But it's the wrong DNA.
What do you mean
I don't
the
care
wrong
about that DNA.
How do you know
Well, it's yours
95%?
wrong. Yeah.
How do you know 95% of it is
Yes,
irrelevant?
let's discuss that issue, because that's...
How do you know that?
Yes. Who
How do you
told
know?
that?
Who told you, or where is the evidence that 95% of the DNA is junk?
It mutates so rapidly between chimpanzees and man, all right, that it's got to be irrelevant. I mean, it's got to be there for spacers. I mean, a guy named Gillespie Davis has studied the globin locus. He studied introns in the globin locus between chimps and humans. And in fact, I called him up and said, well, tell me about intron evolution. And he said, I can't tell you anything. It mutates so fast, we can't even use computer programs to analyze it.
How many genes are there in the worm, Cinerabdus, Sidney Brenner?
So,
How many genes?
right, I'm just telling you that the
95% of that's
introns
junk.
mutate too fast to be important.
It's too fast to be
But
important.
that, it's like saying, as a historian, it's like saying,
Yes.
well, at the end of the 19th century, what's the point of trying to figure out mechanical flight? It's impossible.
No, I don't say that. I'm saying that
It's the same sort of-
everybody makes a
it's
judgment call.
the same
But
sort
it's
of
not a
reason.
judgment call. The point is that science has now shown the first production type sequences have now been done on the worm. That's where the Genome Project decided to start, and that's where the money, and that's what's caused a certain amount of international friction, as some of you will be reading in the newspapers in the next few days. However, the program has now sequenced something of the order of 4 cosmids. That's something like 200,000 base pairs. And much to the surprise and the chagrin of the investigators, the gene density, and by gene density here I should be very careful because we don't really mean gene density. What we mean are open reading frames of nucleotides is 3.5 times the original estimate. Thus, what was once estimated for Cinerabdus of having the order of 5,000 genes, or maybe 10% coding region, is now perhaps going up to 30 to 40%. We don't know until we do the science. Remember that.
There's a question from Tuskegee Institution for Dr. Zinder. What do you think of the patenting of random cDNAs like those of Craig Mentor? Sorry, I just read these.
I think I have been quoted in the various newspapers. I think it should not be allowed. And of course, that's not for me to say. That's for the Patent Office and Trademark Office to make a decision. I do hope that now that it's gone to the PTO, they will very quickly decide to take a decision on it, and on the basis of Section 101, the utility clause will say that this is not useful and will deny the patent. This will clarify the issue. If they do allow patents on cDNAs at this level, one hopes it will be appealed, and ultimately the Supreme Court then will make the decision. However, my own feeling is that one should not be allowed to patent something that you haven't the vaguest idea what it does and can be obtained by almost trivial means.
Okay, a question from the University of Wisconsin. Is there an umbrella organization that oversees the international cooperation efforts of research, development, and ethics of the Human Genome Project?
An international organization?
Hugo.
Is
Isn't
that-
that Hugo?
No, for the ethics portion. Hugo is the whole thing, isn't
Right.
it?
Is there any international effort looking at the ethics of the Human Genome Project?
There's a recommendation to that effect that the structure isn't there to support right now.
There is no...
Here's another related question for Dr. Zinder. You mentioned that the ELSI group was an important goal of the Genome Project. Doesn't it constitute a conflict of interest to have both regulatory and promotional functions served by the same group? Can we believe that the ELSI group will do an honest job?
I don't believe at the moment that the ELSA organization has what we would call regulatory function. It is a means by which support can be obtained for doing studies on the ethics or on cystic fibrosis and so on. So it's a granting agency. It is not a regulatory agency in this area. That is, it will come up with positions and position papers and points, which it would then be the responsible of the various agencies of governments that do have regulatory authority in this area to take up with. The NIH has no regulatory authority almost in any area.
This is for Abby Myers. There's several questions. I'll pick just one. Why does someone who works for the National Organization for Rare Disorders want to see a slower pace in research, which is actually the first step in finding the cures?
Well, we don't want to see a slower pace. We want to see a faster pace. And what's happened is scientists who are studying diseases or groups of closely related diseases were working very hard on finding kindred, large groups of people who are related to each other who would have a disease so that they could try to search for the genes and develop some type of genetic tests. And those clinicians and basic researchers have found it very hard to get funding to continue their individual genetic studies looking at the actual cells of people with these genetic diseases. And they feel that it's because the Genome Project has taken so much money has been diverted so that you can study the genes of the normal healthy person rather than these people who they're trying to target in on. If you look at things like Huntington's disease, the chromosome marker was found because they found a large kindred in Venezuela, and it was from their blood and the blood of other kindreds that they found through the University of Indiana's registry that they were able to find the marker. Now, what happens when these scientists who are studying these specific diseases don't get funded? And their anger is at the Genome Project for taking the money away.
I think we have a question from the auditorium. Go ahead.
Thank you. I inadvertently ended up seated closest to the mic, so I thought I'd ask the first local question. My name is Ron Limoges. I'm a doctoral student here in public policy, and I'd like to shadow a comment made by Ms. Myers about the transfer of the technology. I guess we could debate the technology for the rest of our lives. It's proceeding apace, and we'll learn to live with that. My concern is with the physician transferring that technology appropriately and intelligently to his or her patient. The history of certainly the cosmetic surgery profession over the last few months is an embarrassment to them and to the medical profession as far as I'm concerned. And I'm wondering if we'll see some of the same thing in the transfer of this genetic technology as we did with Joseph's disease and some of the other problems in terms of genetic mapping. Thank you.
Is that question aimed at me?
You're welcome to answer.
Oh, I think that there are lots of companies out there very willing and eager to transfer the technology through some saleable product. For instance, gene therapy. I think that before the year 2000, we're going to see gigantic strides with gene therapy. Now, what is there that is commercially possible in gene therapy? Not the enzymes or the proteins, because those are they already exist. It's the vectors. Designing vectors will be a boon industry, and the other biotechnology drugs that are going to put enzymes back into the human body are also going to make a lot of money. I mean, every biotechnology drug costs a lot of money, and some of them are outrageously high. But there's going to be a lot to sell out of this whole project because as they find the genes and find out what protein or enzyme they don't make, some drug company or device company is going to figure out a way to replace that product.
Okay, here's a question for Dr. Cook-Deegan. You work now in the area of neurological and mental disorders. Wouldn't it be useful to identify people with such problems based on their genes to allow them better or earlier treatment? Or do you see the Human Genome Project as a threat to the well-being of people with such problems?
I don't think there's a simple answer to the question. Perhaps I didn't do a clear job of it. But what I was trying to capture in my introduction is the reason that those of us who are coming at this from human genetics, trying to understand human genetic disease, were so enthusiastic about the Genome Project is because it provided a set of tools to more rapidly find the genes we cared about. So in the case of Alzheimer's disease, we wanted to find those families where we could find a broken molecule that would explain what it is that causes Alzheimer's disease. It doesn't mean necessarily we would have a cure for it, but we would finally have a molecular handle on, in those families, it's that gene that causes that disease, and we'd be able to study it using all the tools of biochemistry and the existing tools in our armamentarium to be able to get at it more quickly. Now, the fact is that the world is moving in this direction. If you look at any institute, the disease institutes, every institute at NIH, the number of grants going to human genetics has been increasing very dramatically over the last decade. The Genome Project, in my view, came out of the technological developments from a rising wave. It's not something that has been thrown out in front and is trying to, the issue of hype is an important issue. People were enthusiastic, they were excited. They did call it the Holy Grail. All these things are important. But when it gets right down to it, you're studying a family with a genetic form of Alzheimer's disease. You want to study bipolar disorder or manic depressive disorder. You want to study schizophrenia. It's clear that there are genetic components that play a role in each of those conditions. It's also clear that they don't explain the whole thing. But the point is, what we do as biomedical researchers is try to grab the tools that we've got at hand to try to understand what it is that we're trying to study. So yes, I really think the Genome Project is a way to do that faster. And the analogy that I would like to make is, essentially, you've got a genetic frontier out there. It really has not been very carefully explored. You've got certain people who say, you're not going to find very much interesting out there, or you're going to find a lot interesting, but it's not the easiest way to get there. But you've got a lot of biomedical researchers lined up at the frontier, marching into that territory. The question is, when do you start building roads, and how much do you spend on roads compared to doing the other things that you want to do?
Can I just follow up on this for a moment? It seems to me that there is a kind of schizophrenia that we bring to biomedical research. On the one hand, we want to study individual diseases, and it's understandable why we want to do that for reasons of diagnosis, therapy, and possibly cure. On the other hand, we also want to study the fundamental processes that underlie all organic processes or functions, ranging from the coding of the genes through development, through what goes wrong, and through the comparison of one organism with another on an evolutionary frame. Now, I think that we ought to be supporting both, and we ought to be supporting the things that connect those two. And fundamental processes that involve physiology and biochemistry are part of that. I don't see why we should choose among all of these things. The important point here is that there ought to be a significant and deliberate investment in fundamental processes that underlie all organic processes and development. And there is nothing more fundamental than the mapping and sequencing of genes, of DNA in all organisms. The technologies that will come out of this project will precisely eliminate the drudgery that gene sequencing now requires, because you stick the DNA in a machine and out comes a sequence of base pairs without having to go through the drudgery that everybody has to go through now. These are the avenues and the vision that the project is pointing towards. It's a kind of long-term capital investment in goods that will be of value to all of biology and not just to a particular disease or particular sets of diseases. We need to have both kinds of investment. It's foolish to choose between them.
May I make one on value? I'd just like to make one last scientific debate, and then I'd really hope we talk about ethics, because I really want to learn, hopefully. But value, how valuable are these sequences? And I said that I didn't think they were so valuable if you didn't know the physiology. I put it to you that they're not valuable, and I've got a business person who might indicate that they're not valuable, and that is a new company starting in Seattle with Lee Hood and a fellow named Gates, I believe. And the company is going to sequence, if put together, the Caenorhabditis elegans genome. They're going to give away the sequences. They don't want to sell the sequences, but they're willing to sequence it all. What are they really selling? They're selling machines to sequence. So I would argue that, wait a minute, here's a company that's actually designed to build machines for sequencing, and it's giving away the sequence. How valuable are the C. elegans sequences? I'm not entirely clear that the sequences per se are that valuable. Maybe it's the building machines. If you get the government to buy a bunch of machines, maybe that's valuable. I would argue that the sequences may not be so valuable as we hear them being valuable.
Okay, well, I'm sorry, Dr. Eckstein, we won't be able to get to any more ethics questions, but I do have a final question for all the members of the panel. I think we'll go around. We'll start with Dr. Zinder. And the question is, what do you see as the most critical challenge that the Human Genome Project presents us with in the next 5 years?
And the most critical problem that the Human Genome Program has in the next 5 years, that it made the mistake 2 years ago of writing a document called the Human Genome Program the first 5 years. And this document was not only written, it was handed to the United States Congress. And in this document, there is spelled out in no uncertain terms what the goals we hope to achieve in these first 5 years. And that is not only the kind of maps that we will have, but the resolution of the maps. The kinds of physical maps that we will have, but the resolution of the physical map. There are real numbers, and the amount of sequence that we expect to be generated in the humans and in the various other model organisms. In 5 years, we're going to have to sit down and show that we were able to do what we said we were going to do. So we have made a promise. We have written it down. It's there available. The Congress has it in front of its face, and I can assure you they're not going to let us off the hook. So that's the problem the Genome Program faces in the next 5 years.
Dr. Nelson?
It's hard to estimate for 5 years, but I would hope within a 5-year period, let us say the maximum number of discoveries of and identification of genes for particular diseases would have been made. And at the same time, the very achievements of the Genome Project would awaken the American populace to the great need for genetic counseling, genetic counselors and genetic counseling centers, and the payments thereof for the thousands and perhaps millions of people who need to benefit from this science.
I would say the major challenge is twofold. First, on the part of the human genetics community and the Genome Project people in particular, that the challenge will be to refrain from making claims that I think will inevitably be exaggerated about what they have found concerning the role of genes in human makeup and human behavior. I am much less optimistic about the ability to identify particular genes for such disorders as alcoholism or schizophrenia, manic depression, and so on. We've seen claims about these things announced in press conferences by scientists. The media absorbs them uncritically, and then people take them for truth, and then a few months later, it's found that they're wrong, except that makes page 40 in the newspaper. These kinds of exaggerated claims need to be refrained from on the part of the scientific community. The other challenge is that the lay people of the United States and the rest of the world ought not to be victimized by not only exaggerated claims, but misinformation about what the Genome Project is, what it's going to produce, and what kind of dangers it poses. It seems to me that what the challenge is to introduce truth and reality and accuracy as much as possible into the discussion of its nature and its prospects.
Okay, Ms. Myers?
The greatest challenge is national health insurance. Because if we don't have it, all the people who need to benefit from medical services because of what the Genome Project is able to learn for us, they're not going to be able to benefit from it, because as soon as they get a diagnosis, they're going to lose their health insurance. So we have to have a major turnaround in the financing of health care in order to benefit from anything that comes out of this project.
Dr. Eckstein?
Well, I'm a critic of the project, so let me say that my major criticism, again, is in the policy area, and fundamentally, I think the American public has to be alert to the fact that scientists, like car salesmen, sell. They sell because they're enthusiastic about their craft sometimes, or they sell because they want bigger labs. They sell for whatever reason. Scientists sell, and politicians sell. And the relationship between media hype and politicians and scientific endeavors is a fragile, delicate, dangerous one. And I want to point that out again, because we don't want a nation whose scientific endeavors are generated by powerful politicians, slick media ads, and a lot of hype. We want technological developments that are based in the concrete, not in television sound bites.
Okay, Dr. Cook-Deegan.
I guess I think what the Genome Project has to do is explain itself over the next 5 years. I personally am a supporter of the project. I'm a supporter of the project because I think it's a quick way to get at what we care about when we do biomedical research. I think we need to do a better job of maintaining this alliance between people who care about the diseases that affect them and the biomedical researchers who do the work. I think the Genome Project is a legitimate vehicle for pursuing that, but I think we need to go out and do a better job of explaining that.
Okay, thank you all very much for your comments, for your reactions to each other's comments. I think it's clear to all of us here, and I imagine in the distant audiences, that we have just begun to learn and consider, and that this is a process that needs to go on from now on. For the Choices and Challenges Project, this is Doris Allen wishing you all good afternoon. Thank you.